Hello, I'm Guy Sauvageau. I'm from ExCellThera. I'm the CEO. Okay, so this is me again. So ExCellThera is really an advanced clinical stage biotechnology company delivering molecules and bioengineering solutions to expand stem and immune cells for use in novel one-time curative therapies for patients mostly with hematological malignancies. So I use this slide basically to show our clinical pipeline, and we mostly focus for now on highly malignant disease, AML, ALL, myelodysplasia, and more recently on multiple myeloma and sickle cell disease. Now, the little box at the bottom of the slides indicate a lot of features of the company. First, we're a cell expansion company. As you will see during the presentation, the product is a very different product, and in fact, it's a unique cell composition, which we've shown in about 40 patients as of today to be safe, to generate no graft versus host disease, although this is an HLA mismatch transplant setting. It's a cryopreserved product now, and now going into outpatient treatment. So a bit of the brief history. The molecule was identified, and our key molecule, UM171, was identified in 2010, mostly in my laboratory and that of Anne Marinier, optimized and now GMP synthesized enough for about 10 million graft expansion. We filed the patent in 2013 in multiple jurisdictions. We've initiated and completed our first phase 1/2 trial in 2018, 22 patients, single cord transplant, and I will show you some of these results. Orphan drug designation last year, RMAT this year, and we're now close to a phase 3 trial that will basically compare this technology with standard of care haplotransplant. Manufacturing is relatively simple, it's fast, and it's small. So it basically uses a single cord unit, again, small cords. We select them with Miltini, CD34 representing about 1% of the progenitor cells in these cords. The negative fraction is frozen, and that's basically the immune system, the T cells, frozen until 7 days later the patient receives the expanded cells along with the UM171 molecule, which is washed out right before being frozen or being injected. So this slide is the typical slide to show that it seems to work where many of the other technologies are failing, whether you're using bone marrow, mobilized blood, or cord blood. And let me just be a bit more granular with the next 2 slides. So these are some of the key numbers with this first clinical trial, which is about to be published. We get very little transplant-related mortality. This is cord blood transplant expected in the range of 30 to 45%. We didn't have a single case of chronic graft versus host disease that requires therapy. And using the famous CRFS, we get 75% of the patients at 1 year, which are disease-free, graft versus host disease-free, and of course, which haven't died of the procedure. Now, if I go into the clinical results more deeply, this is basically what I just said, but with the actual curves. So this is the CRFS compared to standard of care. Although, these are only 22 patients, and, we have to realize that that's few patients to make any conclusion based on clinical data. But I just want to show you that although half of our patient cohort were extremely high-risk diseases, including second allos, refractory ALL, we did get extremely good event-free survival, and now with a median follow-up of more than 27 months. Again, transplant-related mortality is n = 1, even with 40 patients transplanted to date. So this technology, as we see it now, is in fact challenging current standard of care in this field. So this is more definitive data in that they won't change because there's enough patient now to be quite secure on this. Neutrophil, oh, sorry about this. Okay, wrong button. Neutrophil engraftment, 100 neutrophil faster with the technology than what cord blood, peripheral blood, and not statistically significant, but a good trend with bone marrow as well. One thing that was remarkable with these patients was that as we measure it, fever resolution, about a third of them, although a lot of these are PSI-TBI patients, did not develop any fever. They just went through transplant in a way that really surprised us. And we certainly tried to find a reason for this, and that's much better than with the other alternatives. And last but not least, immunosuppression withdrawal was almost at 90% at 8 months and 100% at 13 months compared to standard of care in our center, but that's true whether you're in the US or in Europe. So in other words, these patients are completely off immunosuppressive therapy, and as I said, they don't develop chronic graft versus host disease that obviously requires therapy because they're not on it. So we now understand the mechanism of action of the molecule, and I want to, spend maybe more than a few seconds on this slide, which I think is quite critical. So this slide represents single-cell sequencing of a fresh CD34 purified cord. So this is how the cells look like when we throw them in these UM171 cultures. And in red, we indicate what we call HSPCs, or the hematopoietic stem and progenitor cells. And after 7 days in cultures, as you can see, the color, which is the graft composition, changes dramatically. And we get a lot more of these HSPCs by about 18-fold on average. And these are data from the clinical trial. And this allows for faster engraftment, as I've shown you. Now, we get another strange population, which was not present initially, which are called gut dendritic cells. These cells clearly reduce gut damage by helping to regenerate gut, at least in preclinical models. They also have additional functions that I would be happy to discuss. But what is most striking is a huge 8,000-fold increase in that 7-day culture of these CD34 plus mast cells, which are known, again, in preclinical models to strongly reduce GVHD. And although we don't have that data right now, we believe that they're clearly tolerogenic. So something very special, you cannot expand by 8,000-fold a cell population in 7 days unless you transform something that's not it into it. And that's basically what the mechanism of action is showing us. And again, I'd be happy to discuss this on a one-to-one basis. So there seems to be many cellular population at play to generate what looks to us now like maybe some sort of a very good graft. So the ECT-001, which is basically the bioreactor strategy and the molecule, it allows us to use smaller cord blood unit for greater inventory of cord blood unit to select for. Thereby, it ensures better HLA matching. And let me just give you one example of this. So this scatter plot here, this is total nuclear cells, CD34 cells per kilo. Each dot represents a cord in a Canadian bank, EMA Quebec. And each of the big red dots here represent one of the cord that we selected for one of our clinical trials. And as you can see in this upper box here, for an average Canadian or American, about 5% of the cords in a bank are eligible because of size issue. In other words, 95% of them are not eligible. Using this strategy, we then started to dig more and more to the point now that 50% of the cords we can use and expand in these trials. Now, what's very interesting is that whether we use a small cords or a large cord, we do not see difference in the rapidity of engraftment, meaning that we saturate these cells in these 171 culture. Now, as a result of using 10 times as many possibilities in terms of HLA matching, 85% of our patients are 6 out of 8 allele match versus 55% in for people who basically use cords in this category here. Okay. So what about the clinical program? How does it look right now? So there's two things that has happened. So as part of the first trial, there were two major observations. The first one is that we see much fewer relapse for patients, including some, as I described, who were in fact in palliative care, and that includes some lymphoma. So because of this, we're currently testing in Canada and also at the Fred Hutch in Seattle a phase 2 trial where we're basically examining the ability of these grafts to eliminate refractory ALL and AML and also myelodysplasia. Also, because of the lack of side effects that I described to you before, we're testing in 2 additional trials, one in multiple myeloma, non-myeloablated, and one in sickle cell disease, which is about to open in Stanford for the lack of toxicity of these grafts in diseases that, where toxicity would be unmanageable and intolerable. So clear unmet medical needs, AML, multiple myeloma, sickle cell disease. These are the annual incidents, and basically, these are the markets we're currently targeting. Development platforms. The molecule, through its mechanism of action, has several properties, one of which is published, is that it enhances gene transfer. So there's a trial starting at Stanford, also on sickle cell disease, by Matthew Porteus, basically exploiting the molecule in a CRISPR gene modification strategy to enhance the gene transfer to hematopoietic stem cells. Solid organ tolerization. There's a lot of enthusiasm right now, and especially from a couple of groups, but at Stanford as well, to transplant solid organs in conjunction with bone marrow to generate microchimerism, hence tolerance. And so they've approached us, and we've developed the technology akin to what we did in cord to bone marrow, and it's working pretty well. So we're currently trying to scale up the strategy and bring it to the clinic. Finally, platelet production. The molecule has a unique property of enhancing megakaryocyte progenitor production, and we're currently developing this with collaborators at the MD Anderson. Leadership team, co-founders are myself, Anne Marigny, our head chemist, Peter Zanstra, the head bioengineer who also discovered the fed-batch system used in this strategy, David Millett, our CEO, CFO, who's with me here at the meeting, Pierre Caudrelier, who has lots of years of experience in cell therapy, and he's been instrumental in preparing these clinical trials and in getting a lot of the FDA approval that I just described. And our recent recruit, Sven Assange, who's Director of Manufacturing with lots of experience in bioprocessing. So I end with a few contacts if any of you would like to talk to us, and thank you.